The broadcast is now starting. All attendees are in listen-only mode.
Hello everyone, my name is Tracy Ng. I work on the marketing team here at GenScript, and I'll be your moderator today. We've done 6 live webinars since October. They are all available to watch on demand on our website as well as our YouTube channel. Here is a complete list. Hope you will find them useful. Today is our last one of this series. I just have a few housekeeping items to cover before introducing our speaker. First, today's webinar will be available on demand after the live session as always. We will upload the slide deck as well. Next, we'd love to hear from you during today's presentation. If you have a question for our speaker, please feel free to send it through the question box. We'll be answering questions at the end of the session. If we don't get to your question during today's webinar, we will be sure to follow up afterwards. Now I'd like to introduce Dr. Gyan Stewart-Jones. Gyan is a senior research scientist at the Vaccine Research Center at the National Institutes of Health, USA. He started his career in Oxford University, working on biochemistry and structural biology of HIV reverse transcriptase. Obtained his doctorate in structural immunology at Oxford University and became a group leader there, working on HIV vaccines. He has extended his scope of research to additional viruses and is applying the principles and practice of structure-based vaccine design to tackle numerous disease targets of global importance. So without further ado, I'd like to kick off the webinar by welcoming our guest speaker, Gyan. Over to you.
Thank you, Tracy. So today I will describe how we approach structural biology in the context of generating and designing new vaccines, and some of the recent results and work that we've been doing at the Vaccine Research Center at the NIH in Bethesda, USA. So in recent years, computational techniques have really advanced very fast, and we are now implementing different approaches to the formation of new vaccines. So here we've got a schema showing how computational antigen analysis and design feeds into many different aspects of vaccine design. We do the design, which goes into the purification stage and the characterization stage. Then it's further characterized by our electron microscopes and using X-ray crystallography. This is a synchrotron where we collect the X-ray data, and this feeds back into the computer analysis and design phase that helps us design new proteins that then can be produced and used as immunogens that then are tested in animal models. And then the data goes back into the computational analysis for further understanding. And then with that, we are in a position to move candidates towards clinical trials. So we primarily focus on antibodies as a modality of protection. And most vaccines function primarily by the elicitation of protective neutralizing antibodies. So there are different ways that antibodies neutralize viruses. One is simply by attaching to the surface and blocking receptor, host cell receptor attachment. Another is to prevent viral fusion. And then a third form is to prevent viral egress. And so there are different stages of the viral life cycle that can be arrested by neutralizing antibodies. Viruses have evolved a multitude of different approaches to evade immune neutralization, and the HIV virus is a prime example of several of these strategies of natural evolution. The error-prone reverse transcriptase in HIV allows huge sequence variability in the surface glycoprotein, here shown in white and purple, where purple is the most variable regions of this protein, white being relatively conserved. It also undergoes conformational variation, and so on the virions, there are properly folded pre-fusion, as we refer to them, spikes. There are unfolded or semi-folded versions that are probably non-functional, but nonetheless they expose different parts of this that may form decoys, and also the conserved and highly functionally important variable 3-loop region of the HIV envelope trimer is concealed within the closed form, and is only exposed when it engages CD4. So there's conformational variability. That's another factor that evades viral neutralization. And then another very important component is the glycan shield on HIV and other viruses. So there's a heavy, dense, and big cloak of glycans on the surface, and these effectively block access to the protein surface for antibodies. So I'll go into more detail later on that. So the hypothesis overall is that structure-based design of immunogens presenting broadly neutralizing antibody epitopes will result in the development of protective vaccines. And so the mission of the Vaccine Research Center really is to generate new candidate vaccines that can be protective for a number of different diseases. So conformational, I'll talk about 3 stages of different modalities of engineering, and exemplify them with different viruses. So the conformational stabilization we've found works particularly well with the paramyxoviruses. The glycan shield engineering, I'll show you some results on the engineering of the HIV-1 glycan shield, and I'll talk about some conserved epitopes that are important targets for flu and HIV. So first, the conformational stabilization, and for this I'll refer to respiratory syncytial virus and the parainfluenza viruses. So how do we stabilize flexible proteins? So some proteins are moving a lot and flip into inappropriate conformations for neutralization. So we want to fix these proteins in their neutralization sensitive states. So we use several different approaches. Cavity filling mutations is one. The addition of disulfide bonds and protein modifications is another one. And we can modify the stability of secondary structure through our knowledge of the detailed atomic structure. So here I'm just showing you how we can modify a particular region of a protein by introduction of a disulfide bond. So if the angles of 2 residues are appropriate and the distance between these 2 C-alpha atoms is appropriate, then a disulfide bond could be engineered in. And so we use in silico methods to analyze the appropriateness of these angles and distances. And also there's some tools that online, disulfide by design is another from Wayne State University, and these are quite strong covalent bonds that really fix the position, but are quite dependent on the particular angles of those residues. So then we can also affect the secondary structure by introduction of proteins. Either proteins cause a very sort of rigid region of the polypeptide, or they can in fact disrupt alpha helices, which are quite important if the alpha helical structure is being generated by the transition of one conformation into the other, then that process can be affected by the introduction of a protein that effectively prevents the formation of hydrogen bonding interactions between residues of the alpha helix. And so we've also generated in this laboratory, Peter Kwong's laboratory, through Guayu Chuang, an automated piece of software that allows prediction of where proteins would be useful in that regard. And then we also use cavity filling mutations as well, which essentially repack the internal region of proteins to make them more stable. And one way of doing that is using PIMOL. And if you display the cavities, you can see these kind of blobs here showing the cavities that exist within the core of a protein. And with that information, you can introduce residues that may fill these cavities more effectively. But there are also some programs that can assist you with that. So in PIMOL as well, in silico mutagenesis allows you to test different side chain alterations at different positions, and it will then come up with these red disks where you can see the introduction of a tryptophan here creates a lot of red disks. There's some massive clashes, and this would not be compatible with the original structure. Whereas where there's minimal formation of these little disks, it kind of suggests that we have an alteration that is more compatible with the formation of the structure. So with that, we can guess and triage different mutations. There are a number of different programs that enable you to do this automatically, but using different constraints. BindProfX is one from the University of Michigan. Rosetta is another very common one that David Baker's group came up with. And there's Osprey 3, but there's also plenty of other ones that allow you to test in silico different modifications of interest. So moving to some specific examples of how we've implemented these approaches to the creation of new vaccines. One big problem in the world is respiratory syncytial virus, RSV. This accounts for about 6 to 7% of global morbidity, and is one of the last remaining childhood diseases without an effective vaccine, and comprises quite a significant amount of lower respiratory tract infections around the world, and indeed many children are affected by this. The RSV virion has both attachment proteins, this HN, H or G, and the fusion F glycoproteins. And it was noticed back in around 2013 that there are two shapes of F glycoprotein on the virion, a kind of long version and a short version. And this is the post-fusion form. This is the pre-fusion form. And this is a map of the gene itself, the fusion glycoprotein, which comprises of an F2, an F1, and a region that's actually clipped out of the two when the protein matures. So this is a crystal structure of the post-fusion structure, which was used in a clinical trial and did not show any efficacy. But in fact, one of the very most potent neutralizing antibodies, D25 to RSV, binds this pre-fusion conformation. As you can see, the F glycoprotein is a completely different structure here, and is and this work was done by Jason McClellan, published in 2013 in Science, where the three D25 antibodies lock the pre-fusion conformation of this F glycoprotein in its state right there. And then what we do know is that this is the active form of the fusion glycoprotein as it sits on the virion, and then it transitions to this form either spontaneously because it's a metastable protein, or in the natural function of this F glycoprotein to cause the virion membrane to fuse with the host cell membrane. So this is basically a little machine that, a nano machine one might refer to it, that causes the fusion of the host cell and the virus membranes. So there are a number of neutralizing antibodies that were identified and mapped onto the pre-fusion conformation of this trimer, and they all, well, many of them bind, the most potent ones bind at this top part as you can see here, D25, AM22, and 5C4. And this part is referred to as the site 0 epitope, which is present on the pre-fusion conformation, but not on the post-fusion conformation. There's another site, site 5, that's also exclusively on the pre-fusion conformation, but not on the post-fusion conformation. And what we saw is that also the most potent epitopes, or potently neutralized epitopes, are indeed on this structure, but not on this structure. So the goal from now was to create a pre-fusion stabilized structure of the RSV fusion glycoprotein. And so based on the structure of the complex, or the stabilized F-glycoprotein with the D25 antibodies bound, we found a number of different stabilizing mutations, including a disulfide bond and a couple of cavity-filling mutations that caused this protein to be produced as a recombinant protein in the absence of this D25 antibody. And what we can see is that in mice, as you include more and more of these different pre-fusion F stabilizing mutations, we have an increase in the elicitation of potently neutralizing sera. And as you can see here, the dsCAB1 has the highest neutralizing response, and that's a combination of a number of these different individual mutations. And the same happens in non-human primates, where we have a significant improvement in the post-fusion versus pre-fusion stabilized immune responses. Now this has moved into clinical trials at the VRC and in a number of pharmaceutical companies. So besides RSV, there are a number of other diseases that are causing major medical problems in the wide world. Parainfluenza is another one. And you can see from here an analysis over the course of about 6 years over the United States. There were these waves of outbreaks of parainfluenza virus 3 shown here in green, almost on an annual basis, and on a biennial basis, parainfluenza 1. So these are causing hospitalizations, and the numbers here show that about 10,300 people per year are admitted to hospital with severe respiratory distress, and this costs the economy around 43 million on average over this time course per year. So we applied the same principle of pre-fusion F stabilization and asked the question whether we can actually get a vaccine candidate that protects or has very potent neutralizing antibodies elicited. And so we started off with the post-fusion PIV3 to identify monoclonal antibodies that are potently neutralizing but don't bind this post-fusion conformation. And so we identified these two antibodies, PIA3 and PIA174, in collaboration with the IRB in Switzerland. And we were able to identify the binding sites of these on the pre-fusion PIV3 F-glycoprotein. Again, they bind the apex region, which is the region that undergoes the most conformational change in transition from pre-fusion to post-fusion. As shown here, the homolog PIV5 F pre-fusion structure compared to the post-fusion PIV3 F glycoprotein. You can see the dramatic conformational change. These antibodies, in fact, lock the pre-fusion conformation of the F glycoprotein. And so with that, we were able to triage our designs that were aimed to stabilize the pre-fusion conformation. And we were able to successfully identify about 6 different disulfide bonds that actually do this within the pre-fusion structure, and a couple of cavity-filling mutations that were also very important in stabilizing the stem region, which in itself undergoes substantial conformational change in transition to post-fusion. And so the yields were really quite good. We got about 15 or 10 milligram, between 5 and say 15 milligrams per liter of expression from culture 293 freestyle cultures, and these were then combined. So we had double disulfides, single disulfides, all with these cavity-filling mutations. And one of the double disulfides generated a 23 milligram per liter yield, which was very, very high, and and when we looked at the stability at 37 degrees of the structure over the time course of around 4 weeks, we saw that this particular mutation retained 100% pre-fusion conformation with no antibody bound, of course, over this time frame, whereas a number of other ones somewhat decayed, as you might refer to it, into the post-fusion conformation over this time course. So the best design seemed to be this double disulfide, which generated 23 milligrams per liter. And so we structurally analyzed this design using Cryo-EM with Priyamvada Acharya, and we complexed this very interesting antibody that binds the apex, this PIA3. In fact, it binds right at the top of this trimer in a 1 antibody to 1 trimer stoichiometry, a bit like the PGT145 like antibodies that in HIV trimers. And what we saw is that this antibody inserts a hydrophobic pair of residues into the apex, into the hole right at the top, and it makes contacts to each one of these protomers. The structures of PIV3 and PIV5 are in fact somewhat similar. The homology is relatively low, about 25% sequence identity there. But nonetheless, we were able to see the engineered disulfide bonds by analysis of the electron density. So do these pre-fusion stabilized immunogens generate potently neutralizing antibodies? So we immunized mice with 10 micrograms of protein with poly-IC twice and read the neutralization titers out at week 5. And what we can see is that the post-fusion conformation elicits very weak immune responses, about 1 in 1,000 titers there, compared to all of these different disulfide stabilized pre-fusion conformations, where we're seeing very, very high neutralization, in the order of about 1.5 million is dilution for these different designs, indicating that there's a step function, and that once you've stabilized the pre-fusion conformation, you don't necessarily need to stabilize it further, except for the fact that one of these generated a higher yield, and that might be beneficial in terms of manufacturing. So we can see that conformation is very important in terms of the elicitation of potently neutralizing antibodies, and in this particular case, the titers were extremely high, much higher than anything that exists in humans by natural infection, and so we were pretty encouraged when we saw this data. We applied the same principles to PIV1, 2, and 4 F glycoproteins, and here are some models showing some of the residues that were modified in the efforts that enabled us to stabilize these pre-fusion conformations. And under negative stain electron microscopy, we could see that we had indeed stabilized what looks like the pre-fusion F trimers for these PIV1, 2, and 4. They appear slightly different because they're in different positions as family members in this virus group, and the pre-fusion F structures were actually quite stable over the course of about 1 week as we analyzed them at different time points under the electron microscope. So we then immunized mice similarly to the PIV3 experiment, and found that, in fact, in all 3 of these additional cases, there's statistically significant differences between the post-fusion and the pre-fusion stabilized groups. And although the difference is lesser in the PIV4, it's highly statistically significant for PIVs 1 and 2, indicating that, again, the pre-fusion conformation of these 3 F-glycoproteins is critical for the elicitation of the better, more potent neutralizing antibodies by these particular immunogens. So then we asked the question, can we effectively fill in the neutralization by having several of these immunogens together, but missing one out? So can we effectively fill the omitted virus type by cross-neutralization? So we generated immunogen mixtures of post-fusion for PIVs 1 to 4. Here we have a pre-fusion 1 on its own. The combination in stoichiometric ratio of the PIV pre-fusion 1, 2, 3, and 4. Here we're omitting the 1, here the number 2, the number 3, and then the number 4. And so the question is, can we fill the neutralization with cross-neutralization? So these are the results, and we can see again we're getting good neutralization of the pre-fusion versus post-fusion, comparing the triangles versus the circles, which are all the pre-fusion groups, the triangles are the post-fusion groups, and where we have the quadrivalent mixture shown in these red squares, we can see that we're getting good potent neutralizing responses to all 4 viruses compared to post-fusion. And where we're emitting the PIV1 here, then suddenly we get a reduction in the neutralization potency to lower, in this case, than the post-fusion PIV1. And for PIV2, again, we're getting a reduction in the neutralization titer. PIV3, we're completely abolishing the neutralization titer there where we miss it out. And PIV4 again is reduced down to the level of the post-fusion. So in all, what this tells us is that we actually need epitopes from each of these viruses to be presented to the immune system, and that even though you've got 3 individual components present, the missing one effectively is not cross-neutralized. So the serology elicited by these F-glycoproteins is type-specific. So moving forward, we developed a non-human primate immunization experiment where we decided to combine the PIV 1 to 4 immunogens and immunize them 3 times, either as the pre-fusion PIV 1 to 4 or the post-fusion PIV 1 to 4 in 2 separate groups of 5 non-human primates. And we then read the neutralization out for all 4 at the 18-week time point, and then continued to boost with PIV3 pre-fusion or post-fusion on its own afterwards, and just to monitor how high we can actually go with the PIV3. So again, with the quadrivalent PIV 1 to 3 4 pre-fusion vaccine, we saw significant improvement in the neutralization titers where we had the pre-fusion structures immunized versus the post-fusion structures. And we're getting immune responses that are potent to all 4 virus types. So this is an encouraging step towards the identification of a quadrivalent vaccine that could, in fact, protect against all 4 of these viruses. So there's no interference between the F-glycoproteins in terms of elicitation and neutralizing responses, at least in non-human primates and in mice as well. So this is likely to be similar in humans if we take it forward. And then if we boost further with the PIV3 post-fusion or pre-fusion on its own, then we see further improvements in the neutralization. So before I showed you the PIV3 after the 3 immunizations with the quadrivalents reaches 11,400. After a further boost of 100 micrograms of PIV3 F-glycoprotein, we're taking it up to about 5-fold to around 52,000. And then it seems to plateau a little bit at this level at week 46 after another PIV3 only immunization, it doesn't seem to take it any further up. But if we look at some of the humans that have elicited potent PIV3 neutralizing responses, this is a cohort of donors. I think it's 27 patients are shown here out of around 127, 23 out of 127. And the most neutralizing human serum that was detected elicited a dilution factor of 1 in 1,500. So what we're achieving here is about 30 times higher than is required, more so than the highest human donor. So our vaccine appears to generate immune responses that are substantially higher and more consistent than what is observed in humans. In most cases, the humans are really very, very low, around in the hundreds level or below. And remember, this is a disease that actually is universal, so pretty much everybody has been exposed to PIV3 and indeed the other PIVs during their childhood and subsequent reinfection. And the symptoms are very similar to influenza, so people don't get diagnosed very frequently, but these viruses are quite common in society all around the world. So in conclusion, for the PIV1 and 4 design, which was based on structural engineering of the F-glycoprotein, we're seeing consistent pan PIV neutralization titers of greater than 1 in 2,000 in non-human primates. And then we're seeing a 6-fold boost of PIV3 neutralizing antibodies after immunization. And this is a proof of concept for multivalent paramyxovirus pre-fusion F-glycoprotein vaccines, which can elicit neutralizing antibodies to multiple paramyxoviruses. So in principle, this cocktail could in fact be combined with other paramyxovirus or pneumovirus vaccines, such as respiratory syncytial virus. So moving forward to how we've used structure to look at and understand and engineer the HIV-1 glycan shield, this is a defense mechanism that's evolved and continually evolves on the HIV-1 virion, and essentially presents self-glycans on the virion glycoprotein that mediates fusion between virion membrane and host cell membrane. So here in green you can see the sugars basically coating the protein in purple and white underneath. And prior work has shown that you can introduce a disulfide bond between the GP120 and the GP41 subunits that stabilizes this pre-fusion conformation. In addition, a critical proline 559 mutation appears to break one of the critical helices in the GP41 region and further stabilizes this pre-fusion structure, enabling recombinant forms of this trimer to be expressed and purified and used as tools to understand and advance vaccine research in the field of HIV. So what we did was, because it's such a highly glycosylated protein, this makes it extremely difficult to crystallize. And the heterogeneity of the glycans, as well as their flexibility, prevents, in many cases, crystallization. So we used a lattice that had been identified by Marie Pansera and published previously to essentially build a suspension of trimers in a crystalline lattice using these two antibodies, 35022 and PGT122. And there was even space in this lattice to accommodate a single chain FV of VOCO1 shown here in black. So we managed to grow these crystals, or I got these crystals probably in 2014, and we got high-resolution X-ray data that helped us define the glycan shield. So what we were very amazed to see was that there was electron density visible for these glycans, which were thought to be kind of waving around a lot in the solvent. surrounding the trimer. Admittedly, some of these glycans have been stabilized by the presence of these antibodies, but many of these glycans, for instance, this group over here, were stabilized purely by the interactions between the glycans themselves. So they, as you can see, they're contacting each other, and they're self-stabilizing into these kind of micro rafts or domains of glycans via the head groups. This was published in Cell, and in fact was it was selected as a cover for the edition. We were also interested to see that at the V1, V2 region, at the apex here, there was a kind of a ring structure of glycans. The density was apparent and suggested that there was a sort of minimal energy that caused these glycans to almost come together through various different bonds, hydrogen bonds, water-mediated contacts, and other interactions. And we got similar data for a BG505 clade A trimer and a JRFL clade B trimer. And then if you superimpose all of these refined glycan structures onto each other, then we can see some conserved glycans have these stems, and then these different structures of the head groups depending on the strain. And here they are, all three of them superimposed together. So underneath the glycan shield is the trimer protein itself. Between the three clades, which are separated by about 80 years of evolution, there's a conserved core structure shown here in black, and a more structurally variable or evolutionary adapting region shown here in red. And this is a sequence alignment showing that, in fact, after 80 years of evolution, there are regions that are very, very conserved and other regions that are very, very variable. So the structure of the X1193.C1 clade G trimer was to 3.4 angstrom resolution, and data was extending out to around 3.1. We were able to map the B factor distribution across the trimer and show that some of these loops were higher B factors compared to other regions of the protein. Some glycans were actually relatively stable. Other parts of the glycans were more flexible. And other regions that had never been seen before, for instance, the fusion peptide, this was the first time we had actually seen the electron density for the HIV-1 fusion peptide. That was revealed in the X11 structure, and this interhelical region as well was another region of interest where this protein essentially breaks the helix. And there were some subtle differences, but again, the core was relatively conserved. The individual glycans, we can see sort of different structures appearing on the surface of these trimers, and we define them in different ways, like this stem-to-stem forking, where the two glycans are actually positioned very close to each other. They splay apart because of the contact between the N-acetyl glucosamine, the first of the glycan residues of these two. And then where there's more space between the bases of these glycans or the sequence, then the glycans can touch each other by their individual arms or form more complex arrangements of quaternary engagements of glycans, triple entanglements, as we said, stem-to-branch associations, or stem-to-stem alignments, where we have a slightly greater distance, and then the first two N-acetyl glucosamines come together. All of this essentially forms different modalities of blocking access to the protein surface of the antibodies that would want to essentially neutralize the virus, and also triggering of B cells that would otherwise target the HIV surface. One of the primary factors of broadly neutralizing antibodies that is in common is that they very, very commonly bind the protein surface as well as a piece of the glycan. So they stabilize one of the glycans and use that as part of the energy derived from the interaction to lock onto the HIV envelope. So a combination of protein and glycan recognition is crucial in the case of broadly neutralizing antibodies, because the penetration to the surface is restricted by the shield, and thus these antibodies require to make a space and also open up the shield to access. So here are a couple of molecular dynamic simulations of the HIV trimer. We modeled this, and this is a kind of virtual reality kind of simulation. It's not really measured through. It's not a measurement of matter. It's a computer simulation, so there's going to be error here. But essentially, it shows the range of flexibilities that the protein underneath and the glycans can undertake when they are left free in the solvent. And they sweep across different regions of space, but some of them are more restricted and clustered. Some of them don't move so much, but the ones on the loops really do move around, as well as the ones towards the base of the trimer. They arch down where the membrane would actually be existing over here. So this is more kind of a hypothetical analysis of what the glycans might be doing, which is not totally consistent with our observations of the crystal structure, but nonetheless it gives us some clues as to the range of flexibilities and spatial occupancies that the glycans individually can accommodate. So taking together all of the positions of those glycans, we can map, depending on the type of glycan that we're looking at. This is a MAN5, a MAN7, and a MAN9. We can map in 3D on the surface of the protein the actual sweeping spaces of these glycans, and to show essentially which region is the most heavily protected by the presence of the glycan shields. And underneath here, I'm showing the protein itself and the degree of conservation of the protein shown in red here on this scale. So you can see the very small areas. This is a very minimal region, small glycan. In reality, they're more like bigger glycans like this MAN9, and even complex glycans that are even bigger than this. So you can imagine that the spaces that these antibodies have to penetrate through to get to this conserved region is very challenging for the immune system. And so, this does present one of the greatest challenges in terms of accessibility of potent and neutralizing antibodies and vaccine design. So we started to analyze using that data. If you superimpose a given antibody onto the surface, which of the glycans are overlapping with that antibody? And for instance, the VRC01, which is a broadly neutralizing antibody, clashes with 4 of these glycans, N276, 462, 363, and 197. And we did this analysis for a whole set of different broadly neutralizing antibodies, or categories of broadly neutralizing antibodies, binding the CD4 binding site, the apex, or the GP120, GP41 interface. And so with that information, we decided to remove certain glycans around sites of vulnerability, such as here, the VRC01 clashes with these 4 glycans. So we created engineered trimers that have these glycans removed, and additionally exposes the CD4 binding site, and use those as immunogens to try to get immune responses that have better access to the CD4 binding site, for instance. And so we undertook that study in non-human primates as well as guinea pigs, and with what we refer to as the dGly4 version of the trimer, we get very, very potent immune responses to the CD4 binding site now that it's unshielded. So by removing the glycans around here, we're making the accessibility to B cell recognition and triggering much, much higher. And when we test the neutralization to autologous viruses that also have the glycans removed around the CD4 binding site, we're seeing extraordinarily high titers, over 1 in a million. Some cases, animals were 1 in 7.8 million dilution, which is extremely high neutralization potency. And this indicates that the glycan shield really protects these epitopes from neutralization. So in some cases we got cross-viral neutralization. So these are the viruses that were used for the neutralization readout. And in the dGlyT4 versions, you can get cross-protein recognition and neutralization by the protein surfaces to the autologous and, in some cases, heterologous viruses to the immunogens. But unfortunately, when we put the glycans back on in the wild type, so the WTs here, we're basically showing you that by putting these glycans back on, the site becomes shielded again. And so the sera does not generate neutralizing responses to the wild type in great abundance. In some cases it does a little bit, but it doesn't seem to be a potent response to the fully glycosylated trimers, but it's extremely potent to the autologously deglycosylated versions. Again, if we mix all of these immunogens together so that we had several different strains combined, then we could also get cross neutralization across these different viruses. But these antibodies may not be evolved enough to be able to tackle the presence of the glycan shield. And this is illustrated here by the structural analysis of VRCO1 bound to the glycosylated X1193.C1 trimer, where we can see the protein-protein interface in yellow and in blue, shown here the heavy chain and the light chain in yellow and blue respectively. And the glycans that it actually contacts are here, the three glycans that we can see, 197, 386, and 276. There's another one, but it's actually sweeping into the space from slightly further away. But anyhow, the N276 glycan, in fact, forms what we were very surprised to see was that it forms a very extensive interaction with the light chain, which in previous structural studies was just really in the solvent, bound to protein GP120. In this case, we had the glycan, the 276 present, and it revealed the function of the light chain of VRCO1. And that really is to stabilize the conformation and structure of the 276 glycan, because the 276 glycan is like a gatekeeper to the CD4 binding site. It needs to be held in an open conformation. The CD4 binding site is designed for CD4, not bivalent antibody, and so it has a restricted access. And so by opening the CD4 binding site with the light chain, the heavy chain can then get in there and bind to the conserved parts and bind and neutralize many different strains. So we need to evolve the antibodies that are coming up here, or further down select them by subsequent boosts to improve the response to the wild-type protein, the wild-type viruses with the full glycan shields, and thus try to evolve antibodies by somatic hypermutation to mimic what we see here with VRCO1 and generate protein glycan co-recognizing antibodies that are capable of broad neutralization. And so this is an area of ongoing research. The solutions have not yet been fully achieved, but we're still working on that. So how do we tackle a virus with great diversity, like HIV, influenza, or some other viruses with many different strains? So here I'm giving the examples of flu and HIV. And so the reverse vaccinology approach was very, very useful in identifying key epitopes of sensitivity and vulnerability from humans that are potently neutralizing. B cells are then sorted to bind the two probes of interest to isolate individual B cells, and sequence their B cell receptors to then be able to express recombinant antibodies and clone them and characterize their neutralization potency and breadth. And in the case of influenza, an epitope on the stem was identified as being broadly neutralizing and potently neutralized by a certain class of B cells. And so the details here help us really design immunogens that can target this region. And in this instance with flu, the hemagglutinin region was engineered, so this great big distracting immunodominant head region was modified, and then in multiple generations of designs reduced to essentially the stem region that presented essentially the epitope that I referred to before, showing the stem-directed antibody recognition. And using this as an immunogen, you can then elicit in mice and ferrets very protective immune responses. And this was shown by the Boyington paper in Nature Medicine in 2016 on nanoparticles. And so the next sort of design phase for this was to put it onto nanoparticles, and this was already done in another context. But here is an image of what it would look like on the nanoparticle. This is a ferritin nanoparticle. It self-assembles, and you can link it to the hemagglutinin stabilized stem and create this this particle that essentially can generate hetero-subtypic influenza protection. And if you want more details, this is published in Nature Medicine 2015. And moving to HIV, another very variable virus. It's so variable that you can get autologous neutralization, but heterologous neutralization through vaccination is very limited. So targeting a very conserved region of it, known as the fusion peptide, was one approach. And a few years ago, an antibody was identified that binds and neutralizes in this region, and so was the subject of a number of years of effort to try to generate immunogens that can neutralize via this conserved epitope. And here it is bound to an antibody. At high resolution, we can see the individual water molecules. And so this then led to the development of immunogens that scaffold the peptide. Is a very simple thing, just a N-terminus of the GP41 fusion peptide bound to these different carrier proteins. And KLH, which was very heavily used for peptide elicitation and neutralizing antibodies in many different cases, was used as a carrier protein to present the peptide. And then it was used in a number of immunizations, subsequently with a number of trimer protein immunizations to boost the immune responses elicited by the carrier conjugate. And with that, it was possible to focus the immune response to this very conserved epitope on the HIV trimer that in some instances, some animals were capable, these are non-human primates, were capable of generating very broad and quite potent immune responses to a number of different viruses that have the same fusion peptide at this one position. So this was a breakthrough in terms of eliciting broad neutralization to HIV viruses. And some of the monoclonal antibodies that have been isolated from these animals have also shown that individual monoclonal antibodies can be broadly neutralizing. Up to greater than, in fact, 50% of viruses can be neutralized in cases of some of these individual broadly neutralizing antibodies. So overall, I've illustrated the ways that we've used design to show how we can understand our pathogen better, how to understand the immune response to these pathogens better, as well as to create the optimal immunogens to tackle some of these fundamental problems, be them conformational variability, glycan shielding, or sequence variation based on our structural knowledge. We use computational analyses to design the appropriate immunogens. We then characterize them using the broadly neutralizing antibodies to verify that these epitopes are presented and that the immunogen is well behaved. And then we further characterize it through structural analysis by checking that the mutations are correctly formed. Then we undertake immunogenicity studies in animals to show that we can elicit the same sort of antibodies that we're seeing in nature by the donors who've been able to generate broadly neutralizing antibodies. And so in summary, I've summarized the tools of the structure-based vaccine design toolbox and the way that we can apply these tools to numerous different diseases for the development of novel vaccines. And these strategies, and these are not limited to these three, but the conformational stabilization, which I showed, RSV and the PIV 1 to 4 vaccine candidates, how we've understood the glycan shield of HIV envelope trimer, and how it's been now modified to illustrate how we how the glycan shield is so fundamentally important to the defense of HIV in this chronic infection, and how we're getting around some of the issues to do with sequence variation in a pathogen, a variable pathogen like HIV and flu, and many others. Hepatitis C is another variable virus, and how we can target conserved regions by knowledge of particular epitopes that have been previously targeted, but it may not be limited just to those. So there's plenty of work to do in the future to fully overcome these challenges and to create functional vaccines for some of these more challenging diseases. But we're making headways towards that. We've got great candidates now for some of these important paramyxoviruses, and hopefully, as time goes forward, we'll be able to address some of the challenges with some of these more difficult viruses. So with that, plenty of people at the Vaccine Research Center and beyond have participated in these efforts. So I'm just listing here Peter Kwong and his lab. He's the structural biology chief at the Vaccine Research Center, and directs a lot of this work. John Mascola, the director of the Vaccine Research Center, takes forward a lot of the candidates that are generated in the structural biology section and helps with the analysis of the sera and the overall coordination of the research. Barney Graham, who is well known for his work in RSV, has been behind the formation of the RSV candidates currently moving forward. And then other people, particularly the electron microscopy by Yaroslav Zabowski and others in different core regions of the center, have participated in all of this work. So with that, I will conclude the talk, and thank you very much for your attention.